Hello and welcome to another episode of Odds and Ends Season 3. In this episode, we'll be discussing lima bean land races. Conventional cultivars and land races of a crop species represent the primary gene pool available to plant breeders for hybridization and improvement in the cultivars being bred. There's extensive research into different methods of analyzing the genetic resources of a species. The most common ones are genetic markers, which give the opportunity to measure genetic relationships more precisely than morphological and biochemical markers. This is because they're potentially unlimited in number, and they're not altered by environmental factors, and furthermore can be organized into maps where you can detect different genes in different parts of the genome. The most common forms of genetic markers used to measure genetic relationships are RFLPs and RAPDs. The former is more effective and accurate than the latter. The latter is cheaper. Some of the problems of RAPDs include lower reproducibility of results due to the simple fact that the results can be affected by the concentration of reaction components and the cycling condition. Despite this, however, research done by Dr. Dos Santos in 1994 found that RAPDs and RFLPs are roughly identical in effectiveness when they were used to establish genetic relationships among known cabbage cultivars, which means that when cultivars whose full pedigree was known, alongside their genetic relationship, were tested for genetic relationships between one another using RAPD and RFLP systems, they both reach roughly the same conclusion. Which brings us to lima beans, the species we're discussing. There are a total of 5 different groups of lima bean within cultivation, 3 of which are harvested for their dry beans, and 2 of which are harvested for their bean pods. The dry bean cultivars are derived from 3 main gene pools. The Hopi Blanche gene pool, which ranges from Central America into the United States. The Carib branch, which includes the islands in the West Indies and the Amazon basin of Brazil. And the Inca branch, which ranges from Central America into Peru. Later genetic evidence found by Dr. Baudin and Dr. DeBuck reduced the gene pools to 2 gene pools. The Mesoamerican gene pool, which ranges from the US to Argentina, and the Andean gene pool, which extends all across the equator, as well as Central America and Peru, as well as parts of Colombia and southern Brazil. This is based on more recent studies using genetic markers. Lima bean cultivars harvested for their immature bean pods, like green beans for instance, are separated into 2 groups, Henderson and Ford Hook. The Henderson lineage, based on genetic markers, the Henderson line seemed to be derived from the Mesoamerican gene pool. The foundation for the Henderson cultivars was a single bush found in a land race of lima beans cultivated by the Hopi tribe, Virginia. All Henderson varieties are direct lineal descendants of this specific individual. The Ford Hook variety seem to be derived from the Andean gene pool, with several of them being derived independently from different land races. The goal is to determine the genetic relationship between the Henderson and Ford Hook types, and relate them to different land races, to get a picture to see if the original research was correct. To that end, 65 lima bean genotypes were studied. This includes 4 Ford Hook cultivars, 7 Henderson cultivars, and 54 landrace cultivars. These were analyzed using rapid genetic technology and statistical software, and here are the results. The genetic data put all existing cultivars into 3 specific clusters. The Henderson and the Mesoamerican fit perfectly into the first cluster, and the Andean gene pool formed the other cluster. The Ford Hook cultivars, however, formed a distinct third cluster. The genetic data further codifies the idea that there are 2 primary gene pools, the Mesoamerican and Andean. It also confirms that the Andean varieties are what the Ford Hook varieties were derived from, and the Henderson cultivars are derived from the Mesoamerican land races. Later analysis of specific cultivars, specifically a cultivar called Entry 30, was found to resemble both genetically and physically a primitive version of the Ford Hook cultivars. But are still a significant distance between the Ford Hook cultivars and the original Andean cultivars, indicating that Fort Hook cultivars were not a simple mutation to create a determinate growth habit, but rather involved changes in many parts of the genome. One landrace also compared to a white-seeded variety of a Henderson variety called Jackson Wonder. The genetic analysis showed that both varieties seem to be highly related. Given that they also have similar seed size and color, as well as morphology, this would make sense, and that cultivar is likely derived from that specific landrace. All the large white-seeded variants of lima bean within the United States that have been studied appear to be related to each other through one specific sample of a landrace, whilst other cultivars seem to be more related to the other small seeded accessions. All the Bolivian cultivars seem to be derived from the Andean cultivars. Based on this information, it is likely that crossbreeding between Henderson and Fort Hook varieties will yield greater levels of genetic variation from which to select from, whilst retaining most of the traits looked for in various cultivars. For those interested in breeding new varieties of spring bean. For those interested in making new crosses for dry bean varieties of lima bean, regular Mesoamerican and Andean cultivars and landraces being crossbred together would be useful in developing more variability and genetic improvement. And that about covers everything. Thank you for watching. Thank you for watching this video. If you enjoyed this video, please like and subscribe. If you really enjoyed it and think you can donate, you can do so at Buy Me a Coffee, link in the description below. Thank you for watching.